Hello, this is Iron Nathaniel Plummer.
I'm Khalil Lajide,
This is Nikia Lawrence.
and our fourth group member, Malik Dockery, would not be able to speak today, but
And
we will be talking about cell adhesion. So, first of all, we're going to talk about this cell adhesion and its importance. So, the cell is the building block of life, and such that the similar cells combine to form tissues combine to form organs. A group of organs carry out the same specific that carry out a specific function, combine to form an organ system, and ultimately you have your organism formed. Cells attach or adhere to each other to form tissues by means of cell junctions and cell adhesion molecules. So, cell adhesion plays an important role in the formation of an organism. So, what is cell adhesion? Cell adhesion is basically the attachment of cells to one another or the extracellular matrix to for the purpose of communication, signaling, and cellular proliferation and cellular differentiation. Um, so, cell adhesion plays an important role in the formation of an organism, and you could have homophilic or heterophilic interaction or attachment. The attachment between cells or the cell and the extracellular matrix, as the case may be, could be homophilic or it could be heterophilic. So, homophilic attachment or adhesion is basically adhesion between cells of similar receptors. So, homophilic attachments are specific in their attachments because they only attach to molecules of the same receptor. They only attach to cells of the same cell receptor molecules. So, as we will discuss later in greater details, cadherins are homophilic in their interactions between others with other cells. And next, we're going to talk about heterophilic interactions. Heterophilic interactions are the opposite. They are non-specific in their interactions, and they are basically the attachment of cells or the extracellular matrix with cell receptor molecules that are different from the cells that are being attached. So, cell adhesion, like I said before, helps in communication differentiation. Differentiation is basically the specialization of cells to form a specific function. And we have two types of cell adhesion. We have the cell to cell adhesion, which is basically adhesion between cells, and we have the cell to matrix adhesion, which is the adhesion between a cell and the extracellular matrix.
Cell adhesion molecules. Basically, it has different types of junctions, adhesive junctions, desmosomes, adhesion junctions, tight junctions, gap junctions, and focal adhesions. Um, the space involved between the junctions. Adhesive junctions are cell-to-cell points or places where cells attach to cells function and cell shape. Um, there's also a gap junction and tight junction. Gap junction is intermediate and attached to one another. Tight junctions help connect epithelius and makes it permeable.
So before we move on, basically we have four different groups of cell adhesion molecules, and we have now we have the selectins, the integrins, the immunoglobulin superfamily, and we have the cadherins. So we're going to classify them quickly based on their mode of interaction, like we had explained before, whether they're homophilic or whether they're heterophilic. So the immunoglobulin superfamily and cadherins they have homophilic interactions, and while the integrins and the selectins are heterophilic in their mode of interactions.
Alright, so for the cell junction, for the figure we explained before, the figure basically explains functions of the junction and how they act. So basically, hemo same hemidesmosomes.
Hemidesmosomes.
Hemidesmosomes Basically, hemo means half, and desmosomes that means connected to the basal lamen to lower regions and basal lamen region. Um, and then they have the cantons belt around the apical region right here for the adherent junction. Um, Adhesive junctions, as I said, cell-cell adhesive junctions, and desmosomes, and extracellular matrix.
So this figure right here just basically tries to explain um, a polarized cell. When I say polarized, I mean like they have different domains, and these domains have different functions. So this shows like the apical region, and you have the basolateral region, you have the basement region, you have the different regions that have carry out specific functions when it comes to cell adhesion.
Here we have cadherins. Cadherins is the single pass transmembrane cell adhesion molecule that is calcium dependent. It has a homophilic cell-to-cell interaction through the extracellular domain. There are three types of cadherins, such as E-cadherins, which are found in epithelial cells. We have P-cadherins, which are found in the placenta. And lastly, we have N-cadherins, which are found in nerve systems. So here in the figure, we have basically the effects of cadherins on cell adhesion. Basically, in this study, cultured L cells, which are fibroblast cells, as you can see, they did not, well, not as you can see, but they do not attach to each other. So when E and P cadherins were added to these L cells, and the cells started to produce these different adhesions, P cadherin, which is in the red, and the green is the E cadherins. Basically, these groups of cadherins basically started to attach to one another. As you can see, the green E cadherins attached with the E cadherins, and the P cadherins attached to the P cadherins.
And then here we can see like the homophilic interaction that we spoke about before. We can see that P cadherins only it came together with similar P cadherins to form tissues of to form a specific tissue ultimately form a specific tissue, and the E cadherins would adhere to other cells expressing E cadherins because they're alike, and to form other specific tissues.
So here we have the structure of a cadherin. So you see that the extracellular domain of the cadherin is pretty much similar to a zipper. So it has like a zip-like structure which extends outside of the cell, which then it attaches to the adjacent cell. So in order for this to occur, calcium must bind and stabilize the extracellular domain in order for the zip structure to occur. Then you have the beta-cadherin protein, which binds to the cytolytic tail of the cadherin, while the alpha-cactin binds to both the beta-catenin and the actin skeleton. The beta-catenin is bound to the P120 catenine. It is an adherent junction that binds the tail of the cadherin to the plasma membrane. So the P120 catenine mediates the cadherin stability by making sure that it is secured to the plasma membrane, and it also regulates its Rho activity. So next we have integrins. Integrins are made of two large transmembrane glycoproteins with two subunits, which are alpha and beta, which form a non-covalent heterodimer. Integrins do not directly adhere to the extracellular matrix. The way that they do adhere to the extracellular matrix is by focal adhesions and hemidesmosomes. Focal adhesions are a cluster of integrins that interact with the microfilaments through adaptor proteins. Adaptor protein, which is one is taline that binds to the actin binding protein vincillin. and alpha actin in order to directly bind the actin filaments to the cytoplasmic cell of the integrin. So as you see in the figure, the alpha and the beta subunits in the extracellular portion of the integrin interacts by forming a binding site for the fibronectin. Specifically with the alpha subunit. In the cytosolic portion of the alpha subunit, there is a binding site for the adaptive protein taline, which links the cytoskeleton to the extracellular domain.
So now we're going to try to explain how this links to cancer and metastasis. So we understand how cells grow. So cells grow through the process of cell division, whereby the mother cell divides into two daughter cells. So it's a rapid process. It doesn't take time, and it ensures that cells grow when they're needed in the body. So the body works in such a way that as cells divide or grow when they're needed in the body. So if let's say if cells are not needed to be replaced by worn out or dead cells, new cells are typically not needed to be made. So I don't expect my body to make cells when I don't need cells to be made in the body. However, when cells grow abnormally, even when they're not needed in the body, we could actually call that cell to be cancerous. So basically, cancer could simply be defined as the abnormality in the growth of cells. So they could be benign, where they don't spread to other sides of the body, or they could be malignant. And this is when the cancer cells spread from sites other than the primary site. The primary site is basically the site of where the cancer cell originated from. So metastasis is when cancer cells spread. That you could call that process metastasis. So it is the spread of cancerous cells to different parts of the body depending on the adhesion of cells to each other and the extracellular matrix. So if it's a cell-to-cell adhesion, or if it's an cell-to-extracellular matrix adhesion. So cancer cells, they spread by invading or growing into normal tissues. So the only way that they can do that is if the cells are loosely attached to one another. So this is where our cell adhesion comes in place. So metastasis is dependent upon cell adhesion. So basically, the ability of a cell to actually spread to other parts of the body depends on how loosely the cells are in their interaction. So if cells that are adhere to one another are loosely attached to one another, it's easier for invasions or migrations to occur in that cell. So it's easier for cancer cells to spread into that to those cells. So you could just basically say that adhesiveness is said to be reduced between cancer cells. So now we're going to talk about epithelial to mesenchymal transition. So we know that epithelial tissues are polarized, meaning that they have different domains. They have the lateral domain, the apical domain, the basal domains, and they have different functions. So mesenchymal tissues are the opposites of epithelial tissues in the sense that they are not polarized. So they don't have those different domains. So if the cells that make up the mesenchymal tissues are loosely attached and lack the organization that the epithelial tissues have. So they're not as organized as regular epithelial tissues. So epithelial to mesenchymal transition involves the alteration between adhered cells so that the epithelial cells break down and become loosely attached to one another. When these cells are loosely attached to each other, it makes it actually easier for the cells to be invasive or migratory. The epithelial cells becoming invasive basically means that the